Hi, my name is Yang. I'm from University of Washington. I will be presenting our recent work titled ACE, Explaining Single-Cell Cluster from an Adversarial Perspective. As we all know, individual cells are dramatically different from each other. They are so different, maybe because of their environmental stimuli, or because they are within different cell cycles, etc. To study the diversity of the cells, we typically use a technique called single-cell transcriptome profiling, also known as skRNA-seq. Here is an overview of how skRNA-seq data look like. Typically, from an individual, we collect a population of cells containing a mixture of different cell types. We sequence them, and what we end up with is this large cell-by-gene expression matrix. This expression matrix is often high-dimensional, but it is intrinsically reside in a low-dimensional manifold. Here is an example of the UMAP representation of the expression matrix, where each group is colored differently to represent a distinct type of cells. Biologists are often interested to reason about the semantic difference between these groups, and then ask the question like, what are the genes differentiating between different cell types? Here is a common workflow to perform such differentiation analysis. Briefly speaking, given the expression matrix as input, the first step is to use nonlinear transformation, such as t-SNE, UMAP, or even deep learning-based autoencoder to project the cells into a low-dimensional latent space. Within that space, the cells are then clustered in a data-driven way. And finally, we carry out some statistical hypothesis testing to quantify the specificity of each gene to each cluster. However, we notice some caveats of this workflow. For example, first, when carrying out this statistical hypothesis testing in the latent space, the uncertainty introduced by the non-linear embedding is completely ignored. Second, although the cluster is formed in a data-driven way, the stochasticity in cluster assignments is never taken into consideration. Third, the gene specificity to the cluster is treated individually, without considering the dependency among genes. And finally, only gene enriched in a single cluster will be highlighted as the cluster-specific signature. But what about those depleted genes? Though there exist many methods to tackle this problem, they all suffered more or less the limitations I mentioned earlier. To tackle all these limitations, we therefore propose ACE, which stands for Adversarial Clustering Explanation. Here's the workflow of ACE. Briefly speaking, ACE aims to jointly consider the embedding and clustering in a unified framework. Next, I will explain each step in more details. The input to ACE is simply the single-cell expression matrix, as well as the specified cluster number K. ACE will project the expression data into a low-dimensional embedding by using a deep autoencoder. This step is very helpful, not only to eliminate the noise, but also to remove the batch effect at the same time. It is worth mentioning that at this step, we can in principle use any off-the-shelf embedding methods, such as slicer, SCVI, and etc. In this study, we just use one of them, called Saucy. The resultant embedding are subsequently clustered by using the Kmeans clustering. In order to make the Kmeans clustering interpretable, together with the autoencoder used in the first step, we use a concept called clustering neuralization to reformulate the Kmeans clustering as a functionally equivalent multi-layer neural network. As shown here, Each cluster will correspond to one logit in output. In that way, the cluster assignment from Kmeans clustering will be preserved for each cell. Now, by concatenating the cluster neuralization and deep autoencoder together, ACE is able to attribute the cell's group assignment all the way back to the input gene through the concatenated neural networks. As the name suggested, ACE draws inspiration from the concept of adversarial machine learning, where minimum perturbations are maliciously crafted to mislead a machine learning model to misclassify a given sample. But here, we repurpose the malicious adversarial attack for a constructive purpose. This is an illustrative example in the digit classification setting. Given a digit 8, and it's confidently classified as 8 by a pre-trained deep learning model. Now we ask the question, what are the minimum changes to make so that the model will be deceived and classified as 3? We can simply achieve the goal by disconnecting the top left and bottom left of the 8. This is the perturbation we introduced. We argue that the perturbation we found indicate the key characteristic difference between digit 3 and 8. By following such intuition, we analogously find the minimum perturbation to a cell that alter its current clustering assignment. To this end, ACE optimized directly on changing cluster assignment and propagating the changes back to the gene through the deep neural network. We will consider two variants of this task. The first task is called 1 versus 1 setting, where we change the current clustering assignment to another cluster assignment of interest. And the second task is called the 1 versus rest setting, where we change the current cluster assignment to any clusters. After we find such minimum perturbation, we then quantify the cluster-specific importance of the genes by the absolute value of such perturbations. We will induce a ranking on the genes, in which highly ranked genes are more specific to the group of interest. As a sanity check, we apply S to the Kmeans dataset and show that ACE is able to explain digit transition in a pixel-wise manner. These digit transitions are chosen such that each digit category is covered at least once in both directions. The results show that ACE does a good job of identifying a set of pixels that accurately explain difference between digit pairs. For example, to convert 8 to 5, ACE disconnects the top right and bottom left of 8, which is as expected. It is worth noting that the perturbations introduced by ACE are inherently symmetric. For example, to convert 1 to 7 and convert it back to 1 again, ACE suggests adding and removing the same part of 7. To further evaluate the performance of ACE, we use a reason-reported simulation method, SimSim, to generate synthetic SkyRNA-seq dataset. We first generate 20 causal genes to form the cluster structure, and then simulate many redundant genes in order to adequately challenge the method that aim to detect a minimum set of informative genes. These redundant genes are still informative in the sense that they are weighted sum of random causal genes with noise. As shown in the figure, that both dependent genes and causal genes, of course they are examples, all exhibit similar cluster pattern. To compare different gene ranking methods, we use the J-car distance to measure how well a subset of top-ranked genes capture the overall cluster structure. To be more specific, the J-car distance measures the similarity between a cell's k-nearest neighbor when we use a subset of genes versus using all genes. A smaller Jacob distance indicates that the clustering structure is better retained. We compare ACE against other 6 methodologically different baselines, each of which induce a ranking on genes in terms of group-specific importance, analogous to ACE. For example, DSeq2 is a representative statistical hypothesis testing method based on a negative binomial model. The Jensen channel distance, or JSD, is a representative distribution distance-based method which quantifies the specificity of gene to a cell type. The Global Counterfactual Explanation, or GCE, is a recently proposed comprehensive sensing method that aims to identify consistent difference among all pair of peak groups. The Gene Relevance Score, or GRS, is a gradient-based explanation method that aims to attribute a low-dimensional embedding back to the genes. And finally, the smooth grad and shape are 2 representative feature attribution methods. The dashed line in the figure indicates the Jacob distance by using 20 causal genes. Notably, ACE performs even better than that, suggesting that the method successfully identifies dependent genes that are more informative than the example of causal genes. We next apply ACE to a real melanoma dataset, whose cell labels, malignant versus benign, are not determined by data-driven clustering, but instead by orthogonally copy number variations. There are around 4,000 cells and around 23,000 genes in the dataset. To compare the different gene ranking methods, we introduce 2 performance measures. On one hand, we aim to quantify how discriminative are these top-ranked genes in terms of separating malignant cells. And on the other hand, we want to quantify how diverse are top-ranked genes in terms of redundancy. For the discriminative power of the selected genes, we train a support vector machine-based classifier with an RBF kernel to separate 2 cell groups. Then, the discriminative power is reduced to the classification performance, measured by the area under ROC curve. A larger AUROC indicates that the selected genes are more discriminative. For the redundancy among the selected genes, we measure the average Pearson correlation between all gene pairs as a function of K. A smaller average Pearson correlation will indicate that the selected genes are less correlated due to low redundancy. We compare ACE against other 6 different baselines as introduced earlier. We show that ACE is competitive against the existing methods in both high discriminative power and low redundancy with respect to different K. We next carry out a Geneset enrichment analysis by using the top 50 genes reported by ACE. Notably, the genes reported by ACE in malignant cells exhibit 17 enriched gold terms, with 6 not identified by other methods. We argue that the gene panel, which include both enriched and depleted genes reported by ACE, may capture different aspects of the biological process. Finally, we will briefly introduce the future direction which we are pursuing. One possible extension is to apply clustering neuralization to alternative cluster algorithms. For example, in the context of skyRNA-seq analysis, the Loven clustering is commonly used and may be a good candidate for neuralization. And also, you may notice that currently it's under user's control to choose the ranking threshold of top-ranked genes. A promising direction for future work is to estimate the confidence for the ranked genes in terms of the false discovery rate, with the help of the recently proposed knockoff framework. To conclude, ACE can find the minimal set of genes that best explain cluster and is competitive against existing methods. The selected high discriminative genes can be both enriched and depleted. And it was highlighting that ACE is potentially useful for domains even beyond biology. The open source code is available in our lab Bitbucket repository. Thank you.